Okay. So thank you very much, Alain, for this invitation. It's a true pleasure and an honor to be here and to be able to present the work that we're doing in the lab over the last years on really one question that we focus on, which is how the forebrain, and particularly the neocortex, which controls lots of things, sensory perception, motor behavior, cognition, is wired during development. How do you come up with these kind of beautiful circuits that will basically provide a hard wiring that can then be refined to provide function? So how do you build these kind of circuits? Really functioning relies on the formation of these kinds of connections, specific connections between neurons located in precise positions. And although, of course, experience is a true culture effect of circuits, basically these large and complex circuits begin to be established in the embryo during embryogenesis, and mainly by intrinsic developmental program, genetic, epigenetic cascades that will control the formation, the backbone of these kind of circuits. However, these intrinsic developmental program can be modulated, or rather perturbed, by some maternal environment factor. And this feature is particularly well illustrated by the fact that, for instance, prenatal inflammation occurring during pregnancy, and particularly the second trimester, can have very deleterious effect and is a risk factor for several neurodevelopmental diseases, such as schizophrenia and autism spectrum disorder. So intrinsic programs, but that can be sort of perturbed, modulated by the environment. So during this process, this wiring, embryonic wiring, two cell processes play major roles. Cell migration is going to position neurons in very specific places. And for the neocortex, cell migration is important because the assembly of these circuits relies on a whole choreography of migrating neurons. Projection neurons migrate very locally inside the neocortex, and interneurons, which are inhibitory neurons, essential for the excitation-inhibition balance inside microcircuits, are actually coming from this ventral structure, the subpallium, and tangentially migrate over a long distance. And neocortical circuits will actually rely on the integration of these two migratory streams into this beautiful organization. So cell migration is essential. Position neuron, it assembles circuits. On the other hand, and we've heard a lot of beautiful talks this morning, axon guidance or navigation, pathfinding is going to be very essential to build connections and projections over long distances. So great effort in the field over the past decades now has really allowed to identify environmental guidance cues, and we've heard a lot of slits, netrins, but also EPH efferents and homeoproteins that actually act at the leading tip of these two different cells, either migrating cells or growing axons, to control basically the organization of either moving neurons or forming connections. And this essential work, also with the elucidation of the intracellular events that control this process, basically has been instrumental for us to understand how specific neurons in the brain are guided to reach their target. And we've heard a lot about how this is not so simple today and crosstalks, but nevertheless, we now have a sort of a roadmap, a framework to see how circuits are established. However, we still are looking at this with a relatively fragmented view. And in the lab, our goal really is to try to decipher and understand how all these events are coordinated in vivo, because we believe it's going to be important to understand how the forebrain and the neocortex really wires, but also to grasp how small developmental defects that occur at specific time points can, through cascading events, basically lead to pathological wiring. So we really want to decipher this sort of cellular logic, the Lego of really how you build the circuits during time. And we also want to understand how maternal environments can basically perturb this assembly. So today, what I want to show you is, I want to show you that interactions between cells that are not functionally related, but that are just transient neighbors during this development, are actually going to be a key for the development of the neocortex. And I want to talk about interaction between these migrating neurons and growing axons, and also I will present you some results on interactions between neuronal and non-neuronal cells of the immune system. Okay, so first, interaction between migrating neurons and axons. And we studied this in a specific system, which is the thalamocortical system. So what is it? The system is essential for neocortical functioning. It relays most of sensory and motor information from the periphery to the neocortex. Thalamic axon, thalamocortical axons show a very stereotyped trajectory, and they also have a very specific and refined topographic organization. Different types of thalamic neurons relay different types of information to distinct cortical areas, and also a topographically organized projection inside different nuclei are going to be essential to relay sensory maps. And this is particularly true in the somatosensory map, where you have a different organization of neurons that convey maps up to the neocortical area. And last but not least, very essential corticothalamic reciprocal loop interconnect cortical areas with different nuclei of the thalamus, and these loops are essential for sensory processing and neocortical functioning. So, of course, there's been a tremendous and a huge work trying to understand how these different projections are built. But what we found a couple of years ago, and maybe more than a couple of years ago, is that really the development and the organization of this projection is directly related to a cell migration event. And I will be presenting a lot of this work on this schema, basically summarize this work on these schemas where the neocortex here is on the left and the dorsal thalamus is on the right, to really present sort of the essence of what we found. So pathfinding of thalamic axons to the neocortex actually relies on the migration of a subpopulation of neurons, which we called corridor neurons. And they do so by making a breach inside this non-permissive domain. And in collaboration with Oscar Marin, we could show that permissivity for the growth of thalamic axons is provided in part by neuregulin-1. This internal path here to the neocortex is really a sort of a hallmark of mammalian vertebrates. And what we could further find is that the positioning of this corridor neuron by the midline repellent slit 2 that we've heard about earlier this morning is actually essential to make this bridge, and therefore acts as a sort of a switch between an internal path to the neocortex from a default external path of thalamic axons that we can find in mutants in mice, but also is reminiscent of what happens, for instance, in birds and reptiles. So what we have here is really cell migration can act as a dynamic system to position guidance cues and shape the formation of an important projection. What we also found is that this cell migrating event is really related to the formation of reciprocal connections. And this is actually regulated by a very time-constrained modulation of the outgrowth of cortical neurons via another guidance cue, SEMA3 and plexin D1. And after a waiting period, cortical neurons can actually use their reciprocal projection, thalamic axons, as a track to make this nicely organized and functionally important reciprocal connection. So in this system, basically by just moving or displacing or modulating the positioning of these small neurons, you can actually shift entirely the positioning and the organization of a reciprocal connection that is going to be very important for neocortical and brain functioning in general. So this corridor neuron really provide a highly dynamic and tunable system to organize projection. And what we found is that these cells are not general guideposts for thalamocortical connections, but they're also important to organize the topography. So what exactly do we mean? So different thalamic neurons will project to different cortical areas. And what we found is that corridor cells express different combination of guidance cues, such as SLIT1 and netrin1, and that these cues have very specific and unpredictable combinatorial activity. So what does it look like in real life? So this is a tracing. Here is the thalamus. Here is actually the corridor seen from the top. And you have these axons that all go internally but go to different cortical areas or region, more rostral, intermediate, and caudal, and will target different cortical regions. The corridor is really right here, where the axons fan out as they grow towards the neocortex. And what we found, and this work was done in collaboration with the lab of Guillermina Lopez-Bendito in Alicante, is that corridor neurons express gradients of different guidance cues, particularly netrin1 and SLIT1. And that in this context, these two cues have peculiar activities. SLIT1 really acts as a repellent for all axons, and we could show that in vitro, and now we can confirm this using different microfluidic devices. And netrin1 in this system, when it's presented alone, has no chemotactic activity. However, when SLIT1 and netrin1 are provided together, SLIT1 can actually enable and promote an attractive activity for the outgrowth of rostral thalamic axons. And more recently, the lab of Guillermina was able to show that this growth promoting relies on a cofactor, which is not ROBO3, but which is FLIRT3 and actually directly binds to roboreceptors. So basically, you have a system which fans out different thalamic axons to different cortical regions and finally organize the topography of axons en route to the neocortex. So why is that of any importance? So we tested this basically by perturbing the formation of the corridor of these migrating neurons and scramble the organization of axons en route to the neocortex. And what we could find out is that basically when you do that, you perturb the transfer of a sensory map from the thalamus to the neocortex. So corridor neurons, these small neurons located on the path, are doing very fundamental and diverse function in building connections. They're positioning the tract along an internal-external path, and they're also organizing the topographic organization en route to the neocortex by displaying different gradients of cues, and this is an important feature to convey or relay sensory information, sensory map to the neocortex. So what are these neurons? So this is ongoing fate mapping and genetic studies, but what we found is that these corridor neurons are absolutely not functionally related. Basically, they will give rise to GABAergic projection neurons in circuits that are related to fear behavior. So what we find in this system basically is that as the brain is growing, as the brain is wiring, you have interactions between neurons that just pass by that will happily contribute to their fear circuits, but in the meantime have been recruited to control or orient or shape the formation of thalamocortical, corticothalamic projection, and these interactions are actually going to be functionally very important for the elaboration of the system. So in this system, migration can really act as a true positioning system to orient, shape the formation of these connections. So what about interactions between neuronal and immune cells? And I would like to spend the rest of my talk discussing this with you because we're very excited about this feature, and I think that it's probably going to be very important. So we started this, and we started to work on the role of microglia in this system. So why microglia, and what are microglia? So microglia are macrophage resident of the brain. As good macrophages, they do everything that macrophages should do. They protect the system through a response to injury, to inflammation, and they also have a role in homeostasis of the system. Of course, they have an innate immune response, but there has been an increasing growing literature really showing that microglia have a true role during the normal functioning of the brain, and that they perform a lot of other activities. And basically, they do these activities by using the same cellular mechanisms that they use during immune response, which is secretion of factors and phagocytosis, for instance, of cell debris, entire cells, pieces of cells, or synapses. And a large literature really shown that microglia are important during postnatal remodeling of circuits, on synaptic remodeling, pruning, and promotion. And also they regulate neurogenesis and neuronal survival. And importantly, microglia dysfunction at behavioral or functional connectivity level has really been shown to lead to deficits that are really, I would say, reminiscent of symptoms that we can find in several neurodevelopmental diseases such as autism and schizophrenia. So these cells are clearly having a very important role during the refinement and the remodulation of circuits. But what was really of interest for us is that it has been shown that these cells derive from yolk sac precursors and enter the brain very, very early on during embryonic development. And they do so basically as the forebrain is formed at basically E9 or E10. So that raised the possibility that these cells could have an important function during the wiring of the circuits, and also that they could constitute, by acting as sensor of external signal, that they could basically act as a relay of maternal perturbation onto wiring defects that could occur during embryogenesis. So we tested the role of microglia during embryogenesis, and this is largely the work of Paola Squanzoni in the lab, a very talented postdoctoral fellow. And basically, she started this as a collaboration with Alain Bessis in our own institute, who works on microglia and synaptic transmission, and the lab of Florent Ginoux in Singapore, an immunologist who has developed several tools and approaches to study microglia function and differentiation. So Paola was very struck by this finding, which is that these cells have a very uneven distribution in the forebrain at mid-neurogenesis, for instance at E14.5, as shown here with this reporter line CX301 GFP. For instance, you have accumulations here, you don't have cells in that region, etc. And what really struck Paola is that these accumulations are not related to cell death or to proliferative regions, but rather they are related to specific points of decision along axonal tracts or streams of migrating cells. And for instance, they tightly associate here in that region with growing dopaminergic axons. And this is just to show you in a sagittal section, the same dopaminergic axons coming from the substantia nigra and VTA and getting into the forebrain, and you probably cannot see, but this is just to show you that this association here at the tip of the growing axons is not general along the tract, but is really at this specific position. So association with growing axons. And in the neocortex, what we could find is that basically microglia avoid entering the cortical plate until late stages of neurogenesis and gradually invade the cortical plate starting from deep layers, for instance, layer 6 and 5 at these ages, and only entering upper layers postnatally as the cortex grows. So very dynamic, transient feature, because very rapidly after embryogenesis, microglia resume a very homogeneous distribution, which is in line with their sentinel immune function. We were very struck by this distribution of microglia because basically defects in dopaminergic innervation or the formation of cortical microcircuits had been really associated with neurodevelopmental disease, and we thought it was very striking that we could find a tight association with these cells and these different structures. So Paola went on and tried to assess what is the role of these cells in the system, And basically, she used a cross-comparative approach. So first, what she did is she took advantage of a new technique developed by Florent Ginoux, which basically allows to deplete the brain from microglia from early stages of embryogenesis using an early blockage of the CSF1R receptor, which is required for the differentiation, proliferation, and survival of microglia. And this procedure, as you can see here by flow cytometry, really very efficiently depletes embryonic brain from microglia at mid-neurogenesis, but also up until birth. And this depletion is transient because very rapidly after birth, brains repopulate, and at P7, basically they have the same levels of microglia back into the circuits. You can see also this with, I hope, with immunohistochemistry, where we have a very efficient depletion of microglia, no microglia, basically, in the forebrain during embryogenesis. So that was first model for no microglia, and she combined this with another model, which are PU1 mutants, a genetic model, which basically lacks all myeloid cells, including microglia. So we basically have also another model where we lack microglia during embryogenesis. And she combined this with a model of maternal immune activation, where she injected LPS, lipopolysaccharide, during pregnancy. And this lipopolysaccharide is one of the main components of bacterial membrane, and this procedure basically mimics inflammation occurring during pregnancy, also at mid-neurogenesis. And by combining and analyzing these different phenotypes, she basically looked at what those cells can do in normal brain development and what they can actually induce when they are activated by immune signals. So before this, she checked that, and not surprisingly, the absence of microglia or their activation doesn't lead to any patterning defect. We didn't see any early neurogenic defect or formation in blood vessels. So she went on and looked at dopaminergic neurons, because we found that really microglia have this tight contact and even are in close contact, and we can find pieces of dopaminergic axons inside microglia nuclei when we look at mid-neurogenesis, when they are actually abutted or juxtaposed to axons as they grow. And this double labeling immunostaining was done by Philippe Rostin in our institute. So this suggested that actually microglia could do something, and so we looked at their progression in the different models. So what we found in the depleted and P1 mutants, so in both models where we don't have microglia, we found an exuberance of these dopaminergic axons, here labeled with GH immunohistochemistry, which is really an invasion of axons prematurely inside this ventral to encephalic region. And in the case of maternal immune inflammation, we have basically a reverse phenotype with a very small but significant reduction in the progression of these axons. And at birth, when we look, we find a sort of consistent defect in the progression of these axons. Here it's in control conditions. In depleted animals, we have this sort of overshooting dorsally of dopaminergic axons. They have this reduced extension and a more limited ventral outgrowth of these axons in the case of maternal immune activation. So basically what we find are very subtle but very reproducible and robust defects when we don't have microglia, or when we have an immune activation of these cells. We affect the progression of dopaminergic axons inside the forebrain. So what about neocortical development? And we focused on the neocortex because, as I mentioned before, we have this choreography of neurons and the arrival of thalamic axons, and all these different features sort of occur in the same time. And the arrival, the timing of progression of microglia inside the neocortex was really reminiscent of critical events occurring during the formation of circuits. So basically, we looked at the formation of circuits in this context. And what we found is that we first looked at layering, and we found that the absence or the activation of microglia has no impact on at least large features of cortical layering. And we can see that here with immunomarkers such as TBR1, and we can do that also with CTIP2, where we have deep layers labeled here, and we find no differences in the number or the size of these layers, or with COX-1 for upper layers. We also find no defects in the axonal invasion of the neocortical plate by thalamic axons, so all these features seem to be behaving normally. What we found, however, is a defect in a very specific population of interneurons that migrates from the ventral telencephalon, and which I mentioned before is essential for the excitation-inhibition balance in cortical microcircuits. And these interneurons express the transcription factor LHX6, and normally here in controls, what we can see, so they migrate from the ventral regions, we have an accumulation of these interneurons on both sides of the cortical plate, and basically up until E17 they stay out of the cortical plate, and this has been shown to be important for their dispersion, tangential dispersion, but also for their positioning inside the cortical plate afterwards. And when we deplete or we lack microglia or we have immune activation, we see that this process is from early stages perturbed. We have a premature invasion of a number of LHX positive interneuron in the cortical plate. And at later stages, what we found is that normally in control, these neurons accumulate in layer 6 of the neocortex, transiently here. And what we found in depleted or P1 mutants, as well as during maternal inflammation, this distribution across the cortical plate is less precise. And these can actually be nicely quantified. What we have is that cells invade the cortical plate, but they're less precisely organized in terms of layer positioning. So this is really clear using this marker LHX6, and basically it affects this specific population, and we looked at other interneural subtypes, even interneurons that position in layer 5, such as renin interneurons. We never actually see a similar defect, suggesting that it's really specific of this population. So what happens later on, because interneuron migrate even after birth, and I mentioned that the depletion that we're using basically repopulates by P7, but what we still have is at P7 is we still have an imbalance in the positioning of these cell types. So now at P7, LHX6 positive interneurons are mainly in deep layers, and you have almost like a bimodal, I don't know if you can see bimodal distribution. But what we have in depleted embryos that lacked microglia during embryogenesis is we have an increase in the number of interneurons present in upper layers, and particularly layer 3. And we have similar results with a different imbalance in layer 3 and layer 4 for embryos that were subjected to maternal immune activation. And once again, this is highly specific of this population of interneuron. Do we have a sort of impact on the later developing circuits? We looked at paravalbumin interneurons, because these LHX6 positive interneuron basically give rise to fast spiking paravalbumin positive interneuron and to somatostatin positive interneuron, and we found no defect in somatostatin positive interneuron. But when we look at paravalbumin positive interneuron, we found a very subtle, I don't know if you can see, but very reproducible defect in the distribution of these interneurons, where basically we have an increase in the number of interneurons located in layer 4 and deep layer 3 in depleted or maternally immune activated pups. And another feature that we can observe, and I don't know if you can actually see that, but we've had a hard time to quantify this, is that the network seems to be disturbed in the sense that the distribution of these interneurons is less homogeneous. We see some clumps and some empty spaces that we actually see with a very less regular distribution in normal situation. So once again, in this system, what we see when we don't have microglia or when it's perturbed is we see a subtle but potentially very important impact on a specific population of neurons, which is the positioning of these paravalbumin functionally important interneurons for circuits. So we were quite surprised to see such specific effect on dopaminergic axons and on these type of interneurons, which have been associated with a lot of developmental defects in several diseases. So how do microglia do this? And unfortunately, I'm not going to be able to show you at the cell level what they do, but we've been focusing onto the molecular cascade that actually they recruit to do that. As I mentioned before, microglia in postnatal circuits have been shown to do very different things through a specific signaling cascade. One of them is the CX3CR1 fractalkine signaling. This signaling system has been shown to mediate the dialogue between neurons and microglia, to be very important for synaptic remodeling, synaptic pruning, and particularly mutants in this CX3CR1 receptor actually have defects in synaptic pruning in the cortex and hippocampus. Complement receptor 3 has also been involved in the synaptic pruning, particularly in the retinogeniculate system, where some synapses are tagged by the complement, and complement receptor 3 signaling is supposed to allow microglia to recognize inactive synapses and eliminate them by encapsulating them. And finally, DAP12, TREM2, which are adapter and receptor signaling pathway, have been shown to really control different aspects of microglia functioning. These DAP12 mutant microglia are supposed to be pro-inflammatory, secrete a lot of different factors, and these two genes have been shown, while mutated, to induce Nasu-Okolo disease, which is early symptomatic dementia-associated syndrome. So we looked at this different pathway, at least to get a hint of what signaling system are going to be important for microglia during embryogenesis. And what we found by looking at all these different mice is that basically the progression of dopaminergic axons is perturbed in CX3CR1 mutant mice, and basically we end up with a phenotype which is halfway between control and no microglia situation, and it leads to the same effect on dopaminergic innervation at birth. We also found no impact on the layering, on the cortical formation of cortical layers in the different mutants that we examined. But we did find that the migration, or at least the positioning of different interneuron subtypes and LHX6 positive interneurons is affected in DAP12 and in CX3CR1 mutants. And once again, what we find is that the positioning at birth, here you have layer 5, and precise localization of interneuron here in this cortical plate. What you have is you have a way less precise distribution with more cells in upper layer and deep layers in both DAP12 and CX3CR1 mutant mice. And at P7, we also observe a differential distribution of interneurons in upper layers, both layer 3 and layer 4, and this is also observed in the CX3CR1 mutant, although these two phenotypes are not exactly the same in terms of impact on the interneurons. So basically, microglia modulate or have an impact on this process where interneurons are located in the cortical plate. And if you perturb microglia function, particularly in CX3CR1 mutants and in DAP12 mutants, you can also observe these different phenotypes. And I should stress that this is also associated with a defect of the entrance into the cortical plate in the different mutants. So this is really showing a sort of a new interplay between the development of the immune and the neural system, and also revealing that microglia have a true function already from very early embryonic stages, and that they participate to the formation of these circuits, potentially by modulating the progression of axons and also the positioning of neurons in the brain, and that anything that will disturb this activity, including environmental signals and the fact that in this system microglia can act as true sensors of either maternal inflammation, but also any signals that the mother is exposed to, can have consequences on the way circuits are going to be wired. What was very striking is that the cellular targets of microglia, dopaminergic axons, cortical interneurons, have actually been identified or defective in a number of other genetic models for neurodevelopmental disease that are not related to microglia functioning, suggested that coming from a genetic or maternal factors, you can actually have a convergence onto a similar or conserved cellular targets. Okay, so I hope I have convinced you that really by looking at how cells interact in the embryo, non-functionally related cells, we can actually grasp a little bit more how you can go from a defective gene, basically bridging defective gene and observed behavior of phenotypes, by understanding how these different cell types coordinately or not coordinately integrate the cellular dialogue that exists between these type of cells, and that can basically have large-scale, long-range, and long-lasting impacts on how you're going to be building circuits in the neocortex. Okay, so I want to thank the people who've actually done the work. So I very briefly mentioned the work on thalamocortical, corticothalamic, but basically it has been a lot of the work of Ludmila Lockman, a staff scientist in the lab. And Marie-Dec was instrumental in looking at reciprocal connections between the thalamus and the neocortex. All the work on microglia has been really initiated and carried out by Paola Squarzoni, a postdoctoral fellow, and she's been more recently joined by Guillaume Moller, a PhD student. And all this work really benefits from essential collaborations. So for microglia, I mentioned Florent Ginoux in Singapore, who provided us with the depletion model and a lot of approaches in immunology. Alain Bessis at the eBands provided us with a lot of the mouse models that we're using. Guillermina Lopez Bendito, we did a lot of the topography and slit-neutrin combination work. For the physiology on the somatosensory map that I didn't present, we have a very nice collaboration with Clément Lénat in our institute, and SEMA 3-plex Indy1 Faniman in Marseille. And also I want to thank the funding agencies that allow us to actually work, which is very highly profitable, and be happy to answer any questions.